Hey everybody, let's learn a little bit about CRISPR, this really amazing and powerful new tool that biochemists and molecular biologists have to work with, and it has an amazing amount of uses and promise, and it has some ethical concerns that we should also be thinking about. Um, so let's learn a little bit about how CRISPR works in nature with the bacteria that use the CRISPR system to help give them immunity against viral infections. And we have borrowed this technology from the bacteria. It evolved in bacteria. So in the picture here, there is a sort of faint gray outline that's representing the cell wall of a bacterium. So we have the cell wall of this bacterium, and then in the colored part at the bottom of this, we're looking at a section of the bacterial chromosome. Bacteria have just a single circular chromosome, and this is the CRISPR location, the CRISPR locus in geneticist terms, on the chromosome. And there are a bunch of different genes, and then the region of the locus that gives us the name CRISPR. This is where we have these repeats, the black parts, where you have very similar palindromic repeats of nucleotides. And then between those repeats, some different sequences that we now know are sequences of former viruses that have infected the cell. So what does it look like when the CRISPR system is used to help immunize a bacterial cell against a virus? Well, if we start up here at the top, we see the virus injecting its DNA into the cell. This is what any virus does to any cell that it infects. It's what happens when we get a viral infection. The virus binds to the surface of the cell and injects its genetic material into the cell cytoplasm. And then we know what DNA can do. It can direct the activities of a cell, and that's what viral DNA can do as well. Well, if the bacterium is lucky, that viral DNA will run into a Cas complex. Cas just means CRISPR associated, and there are certain Cas proteins, enzymes, that are able to identify foreign DNA and cut it. And if we chop up the virus's DNA, it's no longer going to be able to infect the cell. It will disable that viral DNA, and it can't do the work that it would otherwise, and that is to sort of commandeer the machinery of the cell to make new virus particles. So once the Cas complex has chopped the viral DNA, it will take a piece of that viral DNA and trim it down to a size of about 20 base pairs or so. And this is a chunk of viral DNA that's now called a spacer. And that newly chopped out viral DNA, that new spacer, will be added to the CRISPR sequence in the bacterium's chromosome. And so it will add this new sequence and then a new repeat sequence on the other side of it. And as new viruses infect the cell, it will continue to add new spacer sequences and new repeat sequences to this CRISPR region. It can just grow throughout the life of the bacterium. And so now what the virus has in this CRISPR region is essentially like a library, a catalog of DNA sequences that are identifying all of the different viruses that have infected the cell in the past. And so if any of those viruses infects the cell again, the bacterium has a record of that and will have the ability to respond more effectively to disable that virus and prevent it from infecting and potentially killing the cell. So that's how the bacterium would sort of immunize itself from this particular virus, this new one that has just infected the cell. Our immune systems work in a different way, and I'm happy to talk to y'all about that another time, but that's definitely another video. Let's move on and look at the second part of this diagram, and this is what happens if that same kind of virus infects the cell again. How is it that the bacterium would have immunity and could defend itself better against that cell? Against that virus. So here we have the same part of the bacterial chromosome, the CRISPR region, and we see that it has the spacer that came from this virus the first time it was infected. And what happens the second time around is there will be a protein that is specifically designed to target this particular virus's DNA. And it is specifically designed to target this DNA because it has an RNA sequence associated with the protein that is complementary to the spacer sequence down here in the DNA. So what the bacterium will do is, if it is infected by a virus, or just at a baseline level almost any time, it will transcribe the genes, at least some of the genes in this CRISPR region, and that will produce RNAs that are complementary to the viral DNA sequences in these spacers. There is a chunk of RNA called a tracer that helps bring together all the components that will make up a mature CRISPR-Cas9 complex. Cas9 is one of the CRISPR-associated proteins, and it is the protein that is designed to chop a viral DNA if it reinfects the cell. And what makes the Cas9 complex special and potentially extremely helpful for molecular biologists is that it is very specific to this virus's DNA because it has this guide RNA that was transcribed from the spacer, which remember was viral DNA to begin with. And this sequence of about 20 bases is very, very specific to that exact complementary sequence on the viral DNA. And the CRISPR-Cas9 complexes with these guide RNAs will, on any DNA molecule that they run across, start scanning it for this sequence, a sequence of DNA that's complementary to the guide RNA. And if it finds DNA that's complementary to that guide RNA, that must be the DNA from this virus that had infected the cell in the past. And what would the bacterium want to do with that viral DNA? Well, it would want to chop it up so the virus couldn't infect and potentially kill the cell. And that's what the Cas9 enzyme complex is able to do. The guide RNA and the tracer RNA together help it find the viral DNA, and then the Cas protein has the sort of molecular enzymatic scissors to chop that viral DNA and to disable it so that it can't take over the cell's function. There's one little bit here that I haven't talked about, and it's, again, for another time or for another video, or just ask me if you're curious, and It says DNA targeting if PAM is present. PAM is an important part of how the CRISPR system works. But it's getting a little bit more into the weeds, but if you're curious, please ask me. I'd be happy to tell you. That's enough for this one. If this made a little bit of sense, I'm going to feel good. If it made a lot of sense, major victory. If it didn't make any sense, come bother me to give you a better explanation. See ya.